The overall goal of this procedure is to develop effective genetic engineering methods for an unconventional yeast species, including construction of transforming DNA, DNA transformation, transformant selection, and targeted gene deletion by homologous recombination.
We've established a highly efficient gene deletion technique in the Yarrowia lipolytica P01G strain with potential applications in biofuel and biochemical production.
The main advantage of this technique over existing methods like PCR-mediated gene disruption is that large amounts of gene deletion cassettes and a wide range of selectable markers are available.
In this study, we report the genetic transformation protocol for the Yarrowia lipolytica P01G strain that is efficient, easy, rapid, and reproducible.
The Yarrowia lipolytica P01G KO70 deletion strain constructed in this study showed a very efficient homologous recombination frequency which enables faster and easier screenings of targeted gene deletions.
The first step in this procedure is to amplify by PCR the LU2 expression cassette from a Y lipolytica expression vector and introduce LOXP sites into the 5' and 3' ends of the LU2 cassette. A BAMH1 restriction site is also introduced in primer 1 for subsequent steps of the cloning process. After purifying the LOXP promoter LU2 terminator LOXP cassette, 3'A overhangs are added to the purified PCR product. The A-tailed PCR product is then ligated into a TA cloning vector to yield the plasmid TLU2.